Okay, thank you everybody for attending today. I think what's going to be a really special seminar. Our guest speaker today is Dr. Matthew Porteous, who is a professor, MD, PhD at Stanford Medical School Department of Pediatrics. He's a leader in the gene editing field as applied to stem cells that I'll come back and just revisit in a moment. He did his undergraduate work, magna cum laude at Harvard College, did his MD and PhD at Stanford. He would then return to Boston, where he served as resident and then the clinical and research fellow in pediatric hematology oncology, Children's Hospital of Boston, at Dana-Farber Cancer Center, as well as Harvard Medical School. He then, as part of his PhD, did work with David Baltimore, Nobel laureate, who discovered a reverse transcriptase, for example. And he himself is a leader in the scientific field. So Matt did his PhD work there at Caltech. And then once he finished his education, then he took an assistant professorship at UT Southwestern in Dallas, where he was assistant professor, then went on as an associate professor and then full professor at Stanford, where he's in the Department of Pediatrics. I'm not going to sort of incurrent, into his time today, just to say that with respect to sort of the stem cell field, whether it's blood stem progenitor cells, whether it's airway basal cells, stem cells, he's really been a leader in terms of developing new methodologies in applying sort of the newer methods of gene editing in our field. Many people in the field, including ourselves, use his protocols that have really allowed for efficient editing of stem cells, for example. He's mentored individuals in the field who've gone on to be leaders themselves. He publishes in high-impact journals, serves on editorial boards of committees. I think an important thing is, and it's rare the individual who does this, who is not only a scientific leader, but he's really, I think, a thought leader. There's been a lot of issues that have arisen over the last decade in terms of the ethical implications of how we apply gene editing, and Matt really has had a significant impact on that. And then also in terms of providing guidance with respect to how to deal with the regulatory agencies in translating sort of the laboratory findings into the clinic. So we just want to welcome Matt to UAB. He'll be talking today about genome editing of tissue-specific stem cells to generate stem cell-based drugs. Thank you.
Thank you, Brian, for that introduction. Thank you all for being such great hosts today and coming to listen to my seminar, which will primarily focus on the hemoglobinopathies and cystic fibrosis. This one. These are my conflicts of interest, the most relevant of which is Kamau Therapeutics, which is the new sponsor of our genome editing trial for sickle cell disease. And I'll update you on the first and only patient treated by HDR to correct the underlying disease-causing variant that causes this disease. There's lots of forms of genome editing out there. The genome editing that we like to use and harness and optimize and apply is homology-directed repair-based editing. It starts with a double-stranded break, and we use the CRISPR-Cas9 system because of its ease of design, its high activity, and actually its high specificity as a way of generating a break in our target site of interest. We then provide a donor template that contains the sequence changes we want to make. And I'll describe to you how we do that, such that the homologous recombinational repair machinery can use the template as and make a copy of it and paste the information from the donor template over the site of the break, thereby repairing the break and introducing these new sequence changes. And so being from Silicon Valley, California, that's Apple C, Apple V. Yeah, Apple C, Apple V. So cut, copy, and paste. And in this way, we can create single nucleotide changes, but we can also introduce large cassettes of genes. Now, this system we apply to engineer cells outside the body. The homologous recombination machinery is active in G2 and M of the cell cycle. So it requires cell division, which makes it highly accessible to manipulating, dividing cells outside the body or ex vivo, but challenging when you think that most of the cells inside the body are rarely dividing at any given moment in time. We use a highly purified, we use Cas9 protein, and we use a high fidelity version that was discovered by IDT. And we complex that Cas9 protein with a synthetic guide molecule that can now be made by a number of different and growing number of companies. After electroporating this RNP complex, we deliver the donor on a non-integrating AAV vector. And what we learned is that electroporation actually assists the transduction of AAV, or what people in the lab call the EAT. The reason we use AAV is it's evolved a mechanism to deliver its single-stranded DNA cargo to the nucleus of the cells while being minimally detected by the vibrant sensors in the cytoplasm to naked DNA. And using this system, we can achieve target integration frequencies in a range of different primary human cell types, as shown here. The reason we still really like homology-directed repair editing is its versatility. So certainly one can make single nucleotide changes, and I'll describe how we've applied that to sickle cell disease. But unlike almost all other platforms, it is proven to be highly efficient and specific in inserting large gene cassettes in a variety of different ways throughout the genome. And today I'll focus on how we can insert a gene back into itself to correct downstream mutations, as we've done for cystic fibrosis, describe how we can replace one gene with another gene for beta thalassemia, and describe how we can insert a gene at the end of a gene to add a new function to that. So giving several different examples of this versatility. And this has now been applied broadly by many different labs throughout the world, and apologize for not being able to include everyone's publications on this sheet. So the first example I want to show is the idea of reverting a single nucleotide or changing a single nucleotide back from a T to an A. And so the disease we wanted to apply this to is a disease well known to this community, which is sickle cell disease, in which every patient in codon 6 of the beta globin gene has a T instead of an A, leading to this polar amino acid veiling at position 6 instead of the charged glutamic acid, leading to polymerization of the hemoglobin tetramer under low oxygen conditions through hydrophobic interactions. This polymerization causes a round red blood cell to adopt this stiff sickle cell shape, giving the disease its name. And these thick or stiff red blood cells cause occlusions, causing the inability to deliver adequate oxygen to distal tissues. In the United States, it's still estimated that a median lifespan for people with sickle cell disease is in the mid-40s, but there are 500,000 births per year, perhaps even more, globally with people with sickle cell disease, most of those in sub-Saharan Africa, where the median lifespan is still the first decade of life. So it's a devastating disease that causes early mortality and a significant morbidity throughout the world. So summarizing a lot of work by a lot of people, we designed a CRISPR-Cas9 system that would make a double-stranded break close to the variation that causes sickle cell disease. We would then provide a donor vector that has several sequence changes, including turning the pathologic T to the non-pathologic A, creating silent synonymous changes in the donor template to prevent the CRISPR-Cas9 from recutting the gene after it had undergone HDR, and then including some silent changes to facilitate full homologous recombination from the cut site to the change we want to make. And in a laboratory tissue culture dish, we could achieve HDR frequencies at the allele level of 60 to 70% across, in this case, 6 different sources of CD34 cells derived from sickle cell disease patients. When we take those CD34 cells and differentiate them into red blood cells in vitro, we find that the unmanipulated cells labeled MOC generate primarily hemoglobin S in this in vitro differentiation system with a small amount of hemoglobin F. If the cells have undergone the gene correction process I just described and differentiated, they now generate primarily hemoglobin A, shown in green, a small amount of hemoglobin S because we have replaced hemoglobin S with either hemoglobin A or we've inactivated the gene itself by an indel. But we also see an increase in fetal hemoglobin. And I'll come to why I now emphasize this. I'll note that the group out of UCSF, Berkeley, and UCLA that are developing another homology-directed repair strategy for sickle cell disease using the same guide RNA that we're using also sees in the mauve, light orange, I don't know what color that is, significant upregulation of hemoglobin F derived from CD34, human CD34 cells that have been engrafted into mice. And depending on the mouse, you see differing levels of fetal hemoglobin being upregulated and differing levels of the HDR hemoglobin A in the dark green. But again, a laboratory and now animal finding that this process also upregulates gamma globin. Now, why am I saying this? Well, I'll get to it in a slide or two. So we took our process and licensed it to a biotech company called Graphite Bio. And in August 9th of 2022, the first and only patient was infused with our own CD34 cells that had undergone the RMP AAV6 system that I just described and followed. Now, the cell dose ended up being threefold lower than our desired target when we used more sensitive apoptosis assays. She had a prolonged need for transfusions and required the use of a thrombopoietin agonist called L-Trombopag, which also happens to be an HSC agonist, but ultimately has engrafted with a 90% cellular marrow and is no longer in need of transfusions or growth factors. She's living an active sickle cell disease-free life. She's had no veno-occlusive events. She's traveling internationally, and she's resuming her career as a documentary filmmaker. And as I said, she's now past her 2-year follow-up. In terms of cell dose, this is a paper related to cord blood transplantation. And what these authors did is show that if they broke probability of engraftment versus the dose of CD34 cells given, whether that's total nucleated cell dose, mononucleated cell dose, CD34 cell dose, or CFU dose, the higher the dose, the higher probability of engraftment. So cell dose is important in terms of probability of full engraftment and the pace of engraftment. So while we use an autologous product, it had been manipulated in a way that I'll show you caused significant cell stress. In August of 2024, she came in for her 2-year follow-up. And on month 24, see her hemoglobin is now 9, which is not completely normal. Her platelet count, but is non-transfused. Her platelet count is 127, again, not completely normal, but safe. Her white blood cell count is normal. Her reticulocyte is mildly elevated, though not as elevated as what we see in the commercially approved Casgevy product. And now we start to get in, so clinically, she feels fine with these numbers. Now we get into some of the interesting molecular data. When she was started on L-Trombopag, she developed clonal hematopoiesis with a variant in the BCOR gene that is predicted to be inactivating on one allele. BCOR is associated with enhanced hematopoietic stem cell, or BCOR mutants are associated with enhanced hematopoietic stem cell proliferation. They're associated with aplastic anemia, and they can be associated with post-autologous transplants. After the stopping of L-Trombopag, however, we see a gradual decrease in this clonal hematopoiesis, which I'll show you on the next slide, is marked by what we see at the allele level in the HBB gene. What is very surprising to us, and the reason I talked about the fetal hemoglobin so much, is that she has very low levels of hemoglobin S at 7.8%, because we've inactivated that hemoglobin S gene either through HDR or an indel. She has an increasing amount of hemoglobin A because her HDR alleles are increasing as her clonal hematopoiesis decreases, but she has remarkably high fetal hemoglobin. And when we do sequencing of her NGS sequencing of her beta globin gene, we see a variety of different alleles. We see 3 different HDR alleles, one from AAV itself, it's shown in the top line, and the bottom 2 lines shown in different shades of blue is HDR that occurred from recombination from the nearby delta globin gene. A patch of the delta globin gene has recombined into the beta globin gene, which corrects or reverts the pathologic amino acid, but also adds some what we think will be innocuous changes to other amino acids. What we see is that she has a -1 deletion allele, so 1 base pair deletion and a 2 base pair deletion that track together and are decreasing over time. And so graphically, these alleles are shown in the following way. When she was on L-Trombopag, you can see that -1, -2 cell grew out. And that's correlated with the B-core clone. We need to do single cell sequencing to show that they are in fact together, but there's really, mathematically, there's no way that they can't be together. And you can see in the light blue, dark blue, and in, and the green, that the HDR alleles are slowly increasing in time, as are some of the other alleles. So we are now left with this mystery of why did small insertions and deletions in the exon of the beta globin gene de-repress enough gamma globin to cause red blood cells to develop enough to make her transfusion independent. And we are continuing to investigate this through single cell analysis and whole genome sequencing, and by canvassing the world for their best ideas of how this might have happened. So one patient, thankfully I can sleep because she's doing well, but also a course that could not reliably be generalized to the thousands, if not tens of thousands or hundreds of thousands of patients we'd love to see receive this type of therapy. And when we look back at our IND enabling studies, we see that the cells that had undergone the gene correction process, labeled GC-HBB sickle cell disease for gene correction at the HBB locus for sickle cell disease, had functional properties that overlapped with the unmanipulated cells shown in blue, but were significantly decreased, whether it was in CFU colonies, whether that was in level of human cell engraftment after transplantation into immunodeficient mice. And so just contrasting the level, the scatter plot of blue here versus the scatter plot of green here. So the green overlaps with blue, but you can clearly see there's a statistically significant difference. So why? Well, in part, our manufacturing process was 5 days, and we know that CD34 cells don't love being in culture. But as this trial was going on, a number of investigators, including the Naldini lab and my own lab, and Rafael D'Amicchio's lab, demonstrated that while AAV can deliver that single-stranded cargo to the nucleus without being detected by the cytoplasmic sensors, the structure of the AAV genome, these double hairpin loops, induce a p53 stress response. And when we measure that stress response as upregulation of P21, a downstream target of P53, versus the amount of AAV in the cell, not what we put on the cell. So for example, we put on the cell an MOI of 20, a multiplicity of infection of 2,500, but we find that there's 200 vector genomes in the cell. So only about 10%, little less than 10% gets into the cell. You can see that there's a direct correlation between P53 activation and the amount of AAV that is getting into the cell. So a path forward is to figure out how can we maintain high levels of HDR, but minimize the cell stress. So I'm going to tell you two different ways where we've approached this. The first is work that was done by Sridhar Selvaraj in a lab, who explored a panel of modern day small molecule inhibitors of DNA PK catalytic subunit. This is a protein involved in the canonical non-homologous end joining pathway. There were old school inhibitors from 20 or 30 years ago that were very nonspecific. But as I said, almost every pharmaceutical company from Merck, M3814, AstraZeneca, Vertex, Bayer, and others have developed modern day inhibitors that are much more specific and inhibit DNA PK more significantly than the related family members. And using this molecule, Sridhar was able to show in iPS cells, or sorry, pluripotent stem cells, that even at low doses of AZD7648 for short periods of time, less than 24 hours, we could achieve close to 100% targeted integration in pluripotent stem cells, making HDR not a rare and uncommon event, but the most common event we see in cells when you get used as RMP AAV6 system. When we applied this to CD34 hematopoietic stem cells or stem and progenitor cells, when we use a guide RNA that primarily creates a +1 indel, what we now call an NETJ guide, the AZD could increase targeted integration from 25% shown in white to close to 70% shown in white. And when we use an MMEJ guide, that is a guide that creates a variety of different indels and something we now pay much more attention to. This is actually the guide RNA we used in our clinical trial. We could increase the HDR from 70% to 80% with almost complete elimination of the indels. So our HDR to indel ratio is 54 to 1. Taking a little detour here and showing the data from an experiment I would have never done, but luckily Sebastian and Sridhar didn't ask me and probably would have done it anyway. So Sebastian Viel was a visiting scientist from France and was studying genome editing for a disease called SAVI, sting-associated vasculopathy of infancy. And he was screening guide RNAs, guide 3 and guide 5, that overlap by 1 base pair. And what he found is that guide 3 generated a high frequency of indels and guide 5 generated no indels. And so our standard workflow would be guide 5 goes into the freezer, never to be seen again, and guide 3 is what we work with. But when you add a donor vector, and this is in pluripotent stem cells on the left and CD34 cells on the right, but the finding is essentially the same, that there was a small amount of HDR, even with this quote bad guide. But when we added the small molecule inhibitor of NETJ, we now saw significant HDR and essentially no indels. So there is something more to be learned about how we pick our guide RNAs depending on what outcome we want to use. Now, going back to the beta globin story, the goal was not necessarily to increase HDR, but the goal was to keep HDR at the same level or higher, but decrease the amount of AAV significantly to decrease the cell stress. And so Li Wen, in collaboration with Sridhar, showed that in 3 different sources of CD34 cells, we could decrease the MOI to 600 and achieve the same frequency of HDR that we got at 2500 without the small molecule inhibitor. And that results in a significant, although not entirely eliminating that P21 stress response. The second approach is an approach that was based on a paper reported by Dan DeRoche and his colleagues, showing that a peptide that inhibits a protein called 53BP1. So 53BP1 is a protein that gets recruited to the sites of double-stranded breaks and marks them in that way. It is thought that it inhibits the processing of the break so it can be repaired by HDR. So it's an inhibitor of HDR. And what they were able to show is this peptide inhibitor of 53BP1 increases HDR. But they delivered this peptide on DNA plasmids and other ways that we didn't feel like could be easily translated into our process. So in collaboration with IDT, who made the peptide for us, Ron showed that we could, just like with the DNA PK inhibitor, we could decrease the amount of AAV from 2,500 to 625 and maintain the still very high frequencies of HDR by adding this I53 peptide. What that results in on the left-hand side is on the y-axis, you want high HDR, and on the x-axis, you want lots of colonies to be formed. So the quadrant you want to be in is in the top right quadrant. And with the low MOI and the I53 peptide, you see that we've moved from the red triangles to the blue dots. So we've moved in the direction we want to move. Now in data I won't show, but the advantage also of using the peptide is it's only around for a very short period of time and becomes undetectable somewhere between 8 and 16 hours post electroporation. So we're not perturbing the repair of double-stranded breaks for any longer than we have to. Both of these methods work well to increase HDR in a wide variety of cell types. I showed you the data in pluripotent stem cells, but it also works in T cells. I'll show you the data in human bronchial epithelial cells. And again, the I53 works in airway stem cells, T cells, MSCs, and other cells. So where we are in terms of the clinical trial, the transfer is now, the trial is now sponsored by Kamau Therapeutics after Graphite decided to pursue alternatives. And we have an open IND and sites open, and we hope to be able to treat a next patient with a revised manufacturing process that we hope will result in a shorter need for transfusions, higher HDR engraftment, and therefore the equivalent, still maintaining that reduction in hemoglobin S, but instead getting more hemoglobin A instead of hemoglobin F. And hopefully I'll have some results to present in 12 to 18 months. Now staying with the hemoglobinopathies, wanted to show how another potential solution to this low engraftment challenge that we and others have found. So this was work done by Kyle, Danny, and Joe, and was focusing on beta thalassemia, in which a cousin of sickle cell disease, in which, let me just, oh, good. Instead of the beta globin gene making a pathologic hemoglobin, the sickle hemoglobin, you're not making sufficient amounts of beta globin. And so you end up with excess alpha globin, causing alpha globin aggregation, and these precipitates cause toxicity during red blood cell development. So the thought is that we could solve two problems in one by knocking HBB into one of the two HBA genes. We're fortunate that nature has duplicated the alpha globin genes. So we have alpha 1 and alpha 2. And I'll show you in the next slide, we could design a system that allowed us to knock HBB into alpha 1, but not perturb alpha 2. So in this way, we theoretically could normalize the ratio of beta to alpha. And so we can see here that using this HBA1 knock-in approach, we could knock in the beta globin gene in somewhere between 30 and 50% of the alleles, using slightly longer homology arms than we normally use. And you know, I actually take heart when I see spreads like this, because it means, well, A, there's room for improvement, but B, it means actually we're doing real human experiments where variability is is part of the nature of using primary human cells. When we were able to obtain beta thalassemia patient-derived CD34 source cells, 34 cells from John Tisdale, and apply this system, and then send the red blood cells that have been differentiated to Vivian Sheehan, she showed that we could increase the beta to alpha ratio to 0.5 to 1. So 0.5 betas to every 1 alpha, which is approximately what is seen in beta thalassemia trait, a healthy condition. And the reason I use that as a healthy condition is that Pete Sampras has beta thalassemia trait, and he won 7 Wimbledons. So I'm okay with it. May we all win 7 Wimbledons. But when we took these cells and transplanted them into immunodeficient mice, remember our integration frequency was up above 30%. If we look in the primary recipients, the engraftment of the gene edited cells is below 10%. So the same phenomenon, because we were using the same high dose of AAV and none of the improvements that I talked to you a moment ago. Now, assuming that this might be a problem, the question then became, is there a way we can give these knock-in cells a selective advantage in some way? And so this is again work done by Kyle and Sophia and Job, and was recently published on Nature Biomedical Engineering, and is based on the genetics of this Finnish cross-country skier and his family. And I first learned about these genetics from Oliver Smithies when he was working in his career on developing gene therapy for the hemoglobinopathies as well. And so Eero Montaranta was winning gold medals in long distance cross-country ski racing by record amounts. And when his Finnish doctors and geneticists got together, they identified first that he had a hemoglobin of 22, which is about 50% higher than the rest of us, which is obviously why he had an advantage in long distance races. And the molecular basis of this was the family carries a truncation in the erythropoietin receptor, such that the negative inhibitory domain is locked off. And so the cells are hypersensitive to even low levels of erythropoietin, thus causing polycythemia. There's no history of cancer or other malignancy in this disease, although, of course, at hemoglobin 22, they're a little bit ruddy. And that is a technical term, clinical term. So we said, well, we have the capability of recreating this truncated EPO receptor using the modern day tools of CRISPR editing. So we could either create an indel near the site of the family stop codon, and then cause a premature stop codon, or we could even insert a GFP cassette that would precisely recreate this family's mutation. And what you can see on the right-hand side here is cells that have the truncated EPO receptor engineered into them proliferate significantly more than cells that don't have that truncated EPO receptor. And that the alleles, or the percent alleles that have the truncated EPO receptor, they start out the same, but as there's enrichment during red blood cell development, they enrich over time. So in theory, this might be a way. So then what we did is we attached the truncated EPO receptor into the beta globin knock-in to give our beta globin knock-in cells a selective advantage. And in vitro, the various different ways that we knocked in the truncated EPO receptor all gave the knock-in cells a selective advantage, as shown by the increase in the number of alleles. Now, we'd like to test this in a mouse model. The problem is we don't see good human red blood cell development in immunodeficient mice transplanted with human cells. And the EPO receptor system in mice and humans is different enough that it doesn't really work in mice. So this is going to take some other sort of work to show animal efficacy of the system. Nonetheless, it's a way, it's a biologic method to give your cells a selective advantage when they might not have that selective advantage. Now, there are other diseases such as the inborn errors of immunity in which there is a built-in selective advantage where low levels of engraftment, such as we saw in patient one, may be sufficient to completely cure them of their disease. Now, in the last few minutes, I want to switch to cystic fibrosis. And this is a program in which we are trying to develop, similar to a hematopoietic stem cell transplant approach to sickle cell disease, is a cell-based approach to treat at least one aspect of cystic fibrosis. And the idea is to develop an autologous cell-based transplantation method for serious sinus disease in patients with CF. And like, our team is not quite as big as the team here, but this is a team-based approach, and full credit to Shriram Vaidyanathan for kicking this project off and moving it forward. He's now running his own independent lab at Nationwide Children's, and Don Bravo, a postdoc and senior scientist in Jayakar Nayak's lab, who've done the bulk of the experimental work in this project. And, you know, thanks to Jeff Wine and his continuous wisdom about the entire field. I should have somebody else come up and present this slide. I'm sure you could all do this better than me, but lots of different mutations that cause or variants that cause this disease. And while it has been said that with the modern day triple modulator therapy, only 10% of patients have been left behind, I'd like to argue, based on work from Shriram and others, that number is much higher. So Shriram and Zach Seller's COVID project was to go in the databases from the Canadian registry and the UK registry, and they identified that people from Southeast Asia and other regions of the world had genotypes that would be predicted to be non-responsive to modulator therapy at a much higher frequency than had been predicted from the prior registries. And this data is consistent with the work from Megan McGarry and Susanna McCauley and their colleagues. And it is estimated now, from this, that in South Asian and other Asian and Black populations, that it's not 10% of patients who are going to be left behind, but it's 30 or 50% of the patients who are going to be left behind by modulator therapy. So this is an audience that still believes in gene therapy, but nonetheless, I think to think that there is even a more urgent need as we think globally about the number of patients around the world and their genotypes around their cystic fibrosis genotypes. So the system we're trying to work on is that we would harvest basal cells from a patient with serious sinus disease, correct their underlying mutation ex vivo, verify that the correction occurred, then take the cell product, embed it into a scaffold, and then transplant the cell scaffold back into the sinus, where we'd hope to see engraftment of the basal cells, the stem cells of the respiratory epithelium, which would give rise to the rest of the respiratory epithelium and create a normal CFTR function. So this is an example where we're going to try to insert the gene back into itself, so it's regulated by the endogenous regulatory elements, so it gets turned on in the right cells at the right amount and rescues almost all of the downstream mutations. Now, as you all know, the CFTR cDNA is too big to fit into a single AAV. So the knocking in using a single AAV just won't work. Rasmus Bach, however, showed that you could get larger payloads integrated through this it's one step from an experimental standpoint, but it's two steps from sort of a mechanistic standpoint. So you make a break, you get AAV donor 1 to integrate. AAV donor 1 has this target site for the original nuclease. So now you cut the integrated AAV donor 1, and that facilitates integration of AAV donor 2, and now you've doubled the size of the integration that you need. And so, of course, the efficiency does drop But when Shahram designed his dual AAV vectors to have this sequential integration process, he could also attach, he had enough space to attach a flag to put on the cell surface. So he used the ubiquitous PGK promoter to drive the CD19 cell surface molecule in which the intracellular domain had been chopped off. So it's simply the extracellular domain of CD19 with no intracellular signaling domain. And cells that had undergone this double integration event had CD19 on their surface, which could then be enriched to 60, 70% purity by magnetic bead enrichment. Now, he just recently published a paper showing that when you add AZD to this process, you increase this double integration frequency significantly. And in unpublished data that he shared with me, if you combine the AZD7648 and this I53 peptide, you can now reliably get 30% or higher integration of the double cassettes into airway basal cells and human bronchoepithelial cells, starting to approach a number where we may not even need to do that CD19 selection. Now we have some other tricks up our sleeve that may even increase this 30% to higher. So stay tuned. What does this do to the physiology? So when we take basal cells from people with cystic fibrosis, run them through this process I just described to you, and then differentiate those basal cells in an air-liquid interface culture, and then measure the physiology. You guys know these curves better. You need to know these traces better than me. So I'm going to just go to the right-hand side and say that the unmanipulated cells have a current, a pathologic current, a CF uncorrected current of this. There's variability in the non-CF controls shown over here in right. But when we do the universal correction system, we now see currents that completely overlap with the wild type non-CF controls. And it doesn't show up here, but there is, it doesn't look like it here, but there is a correlation between percent allele correction and percent current correction as well. So we've gotten pretty far now. We can isolate the cells, we can grow the cells, we can correct not just Delta 508, but all the other downstream mutations for CF. We can show in an ALI culture that we've restored CFTR function, but the elephant in the room is how do you get these cells back into the sinus? And this is where Don Bravo's work really kicks in. So the first experiment she did is she took luciferase GFP labeled upper airway basal cells from a mouse transgenic and conditioned the sinus of a syngeneic mouse either with sulfur dioxide or nasal brushing, and showed that she could get long-term engraftment as shown by light emission in the sinuses of the mice that have received these transgenic upper airway basal cells. And you can see that the radiance over time was stable, and you can see these patches of GFP cells integrating into the sinus epithelium, as well as the various different epithelial cell types, including secretory cells and basal cells. When she measured how efficient this was by looking at what percent of the sinus was engrafted with the transplanted cells, it ranged between 20% and almost 50% of the respiratory epithelium. So not 100%, but a number that is within range for thinking you might have a meaningful clinical impact on patients. So this is a syngeneic mouse-into-mouse study. To do the human-in-a-mouse study, we wanted to get away from Matrigel, which is what the other studies have been done with, because Matrigel is not, the regulatory path to using Matrigel in humans would be complicated. And Shahram identified several different matrices that these basal cells grow well in. And given that fibrinogen is already an FDA-approved drug, we identified fibrinogen as the gel we'd like to move forward with. And so in this case, Dawn took luciferase labeled human bronchiepithelial, upper airway basal cells that have been labeled with luciferase by knocking luciferase into the beta globin gene and transplanting these cells into immunodeficient mice and asking whether we see prolonged light emission from the sinus of these mice. And you do see prolonged light emission. They're not super bright, but they're there. And so, and then if she takes cells that we manufactured in which the CFTR cDNA had been knocked in and we have the truncated CD19 cell surface marker, she can see by immunofluorescence that there is patches of human cells, including EpCAM positive cells, CD19 human cells, Q80 human cells, throughout the sinus epithelium of these mice. And then, and here's just a high-powered view of a picture that I can understand. So here we have in white the human KRT5 positive basal cells with human cells above it, and then this human patch embedded in a patch of mouse cells surrounding it. So seeing stable, at least up to 2.5 months post-transplant of human epithelium in the mouse sinus. And excitingly, we actually see in the human cells transplanted by immunofluorescence expression of the CFTR in a small number of the cells that were transplanted. So we see not only engraftment, but differentiation and expression of the CFTR transgene from a donor who had cystic fibrosis. So what are our next steps? So we had a pre-IND meeting with the FDA, asking them questions about what we planned and what they expected for us to do. They, of course, wanted to see us do process development and scale up and optimize the manufacturing process. As I mentioned, we hope we can get the HDR frequency high enough that that second step of NGFR enrichment might not be needed. That remains to be seen. The FDA has provided reasonable guidance on how they want you to look at molecular genotoxicity, and we've carried out a number of those different assays. We asked whether we could follow what had been done in the hematopoietic stem cell field and the development of small molecule modulators, which is to use in vitro studies for efficacy, but animal studies for safety, and not need to perform large animal models of efficacy. And they gave us a concurrence that developmental path was reasonable. They asked us to do a toxicology study, and they asked us to do a biodistribution study. That is, we know you're putting the cells in the sinus, but where else do they go? Do they distribute to the other sinus? Do they do they distribute down to the lower airways? More concerningly would be, do they end up in the kidney or liver or another tissue? And then finally, they asked us to do a feasibility study to prove that my surgeon, Jay Karnak, can actually do what we're proposing, which is in an operating room like setting, can he thaw cells, complex them with a fibrinogen, debride a sinus, and implant these cells into the sinus? And in a sinus that is of similar size to a sinus in a person with cystic fibrosis. And so he identified the rabbit as the model, and we're going to have to immunosuppress the rabbit so that they don't immediately reject these cells. So we hope to initiate those feasibility and biodistribution studies in the next 6 to 12 months. So in summary, multi-directed repair has been validated to insert large gene inserts across a range of loci in the ex vivo setting in primary human cell types. Talk today a little bit about the challenge or maybe the near impossibility of harnessing HDR at high frequencies in vivo, where most cells are not dividing. And it has tremendous versatility. I am just saying what everyone else says, and rightly, is that there is knowledge that comes from the brave volunteers in human phase 1 trials that there's absolutely no animal model can replicate. And we learn so much from their bravery. We now have demonstration that HDR edited HSPCs can graft. It is at a level that may not be sufficient for many diseases, but might be good enough for others. We hope we have identified improvements in our manufacturing process that will allow more rapid and increased engraftment of HDR edited cells, such that the patient reaches a better outcome in a faster period of time. And I think continuing to understand the HDR process, optimal methods of culturing cells, integrating biologic selection methods will lead to further improvements. So with that, I want to thank you for the invitation and the opportunity to present. I get to work with a fantastic group of graduate students, MD, PhD students, postdocs, senior scientists in my lab, my clinical collaborators in this division of stem cell transplantation and regenerative medicine, my colleagues in the GMP facility, and academic and bio and private colleagues around the world with funding sources from a variety of different sources. And then just a picture of some of the people in the lab. They're all smiling in these pictures because they've been fed. So those of you who run labs, keep your lab fed. They tend to smile and work harder. And I'm happy to take any questions if you have any. Thank you very much.
Hey, this is Dr. Raj. I'm online. Can I ask a question?
Sure, I can hear you.
Yes. Hey, first of all, really wonderful presentation, lots of advanced information, and I could just grasp a little bit of what you said. But I have some basic questions. Just let me know if I'm totally out of line here. I think you mentioned that uh, efficiency limitations do exist with the CRISPR product that you use successfully in your patient. But I'm surprised that you were able to get so much of in vivo efficiency. Um, and then you made a comment that there was P53 activation, but isn't that a naturally expected response from double-strand breaks caused by CRISPR-Cas9? And the third one is you mentioned about small molecule and peptides to decrease P21 activation. I don't know if I understood that correctly. That's more of an in vitro manipulation, correct? Which will again further impact engraftment. And fourth question is you mentioned about BCOR mutations in that patient. We all always worry about BCOR mutations because they are found in MDS and AML. So are you linking the P53 activation story with the BCOR mutations, or are they P53 independent? So too many questions. Sorry, and if I'm
No,
totally out of line,
those are all
please.
great questions. I'll see if I can get to them all. So, so when we discovered the BCOR variant, I'll try to take them in reverse order. Reached out to all of our colleagues, experts in the world who study clonal hematopoiesis and BCOR as a cause, and they were very reassuring that while BCOR is associated with MDS and AML, it is never the first mutation. It is always a second or third mutation. And one wrote that it would be unlikely for this clone to transform, and the other wrote back and said exceedingly unlikely. Now, does that mean we stopped worrying about it? No, and that's why we're glad to see the clonal hematopoiesis slowly decreasing over time. In terms of the I53 and DNA PK, yes, they are manipulations that are done in the ex vivo manufacturing as a way to enable, as you said, the reduction in the P53 stress response. We for both approaches, though, because they would perturb DNA repair more generally in the cell, our target is to use those for as short and as low concentration as possible. And so that's where we've done. What I didn't say is that we have collaborated with Giondo Turciano about what is the scope of potential increase with genotoxicity with the DNA PK inhibitor. And while there is some increase in off-target indels and some increase in large deletions at the site of the break, there is no increase in translocations or other signs of genotoxicity. In terms of the I-53 peptide, we actually see no evidence of increased genotoxicity. It looks very clean. Shoot, what were your first two questions?
I talked about the efficiency limitation and how you were able to successfully achieve in vivo in your patient.
Yeah.
And then that P53 activation is a natural response to double-strand breaks.
Yeah. So definitely P53 activation is a natural response to double-stranded breaks. The Nalthini lab has also shown that when you have a highly specific guide RNA delivered for a short period of time, that activation is minimal. It is why, though, our control is to use control for P53 activation compared to RNP alone, which would account for that. So we're not quite down to RNP alone. We know RNP alone treated cells can engraft, that's Casgevy, but it's also the Vor pharma product is an RNP alone, and those engraft even faster than Casgevy. It's a knockout of CD33. In terms of the high levels of engraftment, it's because I think the answer is because we're using busulfan myeloablation. So like all of the other major gene therapy, gene editing trials for sickle cell disease and beta thalassemia, we're using single agent but full dose myeloablation of busulfan such that the residual or the remaining hematopoietic stem and progenitor cells are wiped out. And so engraftment has to come from the drug product or the genetically engineered cell product. That's why we see such high levels. One day, of course, we'd love to get rid of busulfan. Yes.
Yeah. Thank you.
Last to think about it. Excellent.
Yeah.
So my question is, for your clinical study in CF patients, what would be your nasal disease readouts? And then what are the major pitfalls in terms of, because the lung would still have all the inflammation and all those things. So do you worry about false negatives, and then how do you handle it?
Yeah, so we don't have to, we have obviously for our pre-IND, we had to have a clinical trial synopsis identifying loosely exclusion and exclusion criteria. It's not the IND itself, so we can change them. So, again, Cole's the Newcastle here. People who've undergone a lung transplant are not currently eligible for Trikafta or modulator therapy, but they still have sinus disease. So we think those are candidates for this type of approach. We also think the 5,000 and maybe even 6,000 people who could be on Trikafta who aren't might be eligible if they have serious sinus disease. In terms of monitoring, we like the sinus. We talked earlier about this because it's so accessible. Jayakar tells me he can bring somebody in the office, numb up their nose and look at it and take some and do some nasal and do some potential differences quite quickly in an office setting. So we think that's, and of course, what we'll do is we'll put the cells in one sinus and not the other, and hopefully see, well, we talked earlier, ideally we'd see some migration. I don't think that's going to happen, but we'll see, or maybe migration will happen over time. So looking at it that way. The other advantage of the sinus, because it can be easily monitored, if, for example, we saw a polyp form, which would be bad, well, it could be easily removed and we could destroy the product quite quickly. In terms of the, complications of, the lungs, well, this is why you do phase 1-2 trials. We'll see what we see. In the lung transplant patient, one might hope that if they are having challenges in their transplanted lung from serious sinus disease, perhaps this might even help them and protect their lung. Yeah.
It's great talk here. So I just have a question. So I don't experience of mice, but I did like wild type rabbits, and
Yeah.
I got a protocol from from Dr. Waters from University in Australia, Sydney, and we implanted the rabbit septum. And my experience is that epithelial cells, they regenerate, but subepithelial layer is not. They're all fibrosis. Like, but mouse doesn't really have some mucosal glands. That subepithelial is very lacking.
Yeah.
My question is, what's gonna happen is like subepithelial, they're still all fibrotic, like how can you just, that tissue is gonna be, even though Jaker's gonna implant in the sinus cavity, so because of a lot of gonna be still fibrotic or still there, so there will be some type, it goes above the basal cell, but below the basal cell is still fibrotic, something, so that's the questions that, is that really that findings that can we really recapitulate into the human cell?
Yeah, so I'm gonna have you talk to Jekar. So I think what the FDA was really asking us is, yes, we will look for 30 days following to see if we get any engraftment, but this study is really about, can you simply thaw cells, mix with fibrinogen, debride a sinus and inject in an OR type setting rather than the actual biology of the engraftment. But we are going to immunosuppress the rabbits so that if there's a period for transient engraftment, we might see it. But I'll put you in touch with Jayakar.
Yeah, do you know what, when you do the ex vivo knock-in of the CFTR?
Yeah.
You know the off-target integrations?
Yeah, so it's below 0.1%. So we don't and so that's our limit of detection. Does that mean it's not going on elsewhere? No, but it's below our limit of detection.
And do you have a way to select the cells that are devoid of those?
No, we don't. We don't have a way to counter select against the very rare number of cells that might have an integration of either AAV1 or AAV2 that might have gone elsewhere in the genome. But again, the random integration frequency is quite low. And, for better or for worse, right now, we have not seen genotoxic effects from random integrations of AAV in contrast to, say, lentiviral and gamma retroviral vectors. But again, this is a phase 1 safety study and we'll be monitoring for any clinical safety.
Another question, is it possible to use a lentivirus that have a higher load capacity?
Yeah, so I was talking with Brian and others today. People have used, you don't want to use an integrating lentiviral vector because it'll integrate before it'll serve as a donor.
But you can make a non-integration.
Yeah, so if you use a non-integrating or an integrating integration defective lentivirus, some have had success with that. We have not. We've had challenges with transduction and manufacturing of IDLVs. They don't manufacture as well and they don't transduce as well, but others have had some, and I think that is an alternative. But we're so comfortable with AAV now. And honestly, I didn't mention this, but in the ex vivo setting, in contrast to the in vivo use of AAV, the cost of the AAV is like 7th on our list because it's just, we don't use very much because we're not having to distribute it around the entire body.
Thank you very
Thank
much
you.
both for your talk and